Hello dear students, in this video we shall learn about factors affecting the rate of enzyme action. The functional specificity of every enzyme is the consequence of its specific chemistry and configuration. Any factor that can alter the chemistry and shape of an enzyme can affect its rate of catalysis. Some of the important factors that can affect the rate of enzyme action are concentration of enzyme, concentration of substrate, temperature, and pH of the medium, and inhibitors. The rate of reaction depends directly on the amount of enzyme present at a specific time at unlimited substrate concentration. If the amount of enzyme is increased by twofold, the reaction rate is doubled. By increasing the enzyme molecules, an increase in the number of active sites takes place. More active sites will convert the substrate molecules into products in the given period of time. However, beyond a certain limiting concentration, further increases in enzyme do not affect the reaction rate. 2. Substrate concentration. At low concentration of substrate, the reaction rate is directly proportional to the substrate available. If the enzyme concentration is kept constant and the amount of substrate is increased, a point is reached when a further increase in the substrate does not increase the rate of the reaction anymore. This is because at high substrate level all the active sites of the enzyme are occupied and further increase in the substrate does not increase the reaction rate. Temperature. The rate of enzyme-controlled reaction may increase with increase in temperature, but up to a certain limit. All enzymes can work at their maximum rate at a specific temperature called as optimum temperature. For enzymes of human body, 37 degrees Celsius is the optimum temperature. Heat provides activation energy and therefore, chemical reactions are accelerated at high temperatures. Heat also supplies kinetic energy to the reacting molecules, causing them to move rapidly. Thus the reactants move more quickly and chances of their collision with each other are increased. However, further increase in heat energy also increases the vibrations of atoms which make up the enzyme molecule. If the vibrations become too violent, globular structure essential for enzyme activity is lost and the enzyme is said to be denatured. pH value. Every enzyme functions most effectively over a narrow range of pH known as the optimum pH as shown in table. A slight change in pH can change the ionization of the amino acids at the active site. Moreover, it may affect the ionization of the substrates. Under these changed conditions, enzyme activity is either retarded or blocked completely. Extreme changes in pH cause the bonds in the enzyme to break, resulting in the enzyme denaturation. Inhibitors. An inhibitor is a chemical substance which can react in place of substrate with the enzyme but is not transformed into products and thus blocks the active site temporarily or permanently. For example, poisons, like cyanide, antibiotics, antimetabolites and some drugs. Inhibitors can be divided into two types, irreversible and reversible. Irreversible inhibitors. They check the reaction rate by occupying the active sites or destroying the globular structure. They occupy the active sites by forming covalent bonds, or they may physically block the active sites. Reversible inhibitors. They form weak linkages with the enzyme. Their effect can be neutralized completely or partly by an increase in the concentration of the substrate. They are further divided into two major types: competitive inhibitors and non-competitive inhibitors. Competitive inhibitors. Because of the structural similarity with the substrate, they may be selected by the binding sites, but are not able to activate the catalytic sites. Thus product are not formed. Non-competitive inhibitors form enzyme inhibitor complex at a point other than the active site. They alter the structure of the enzyme in such a way that even if genuine substrate binds the active site, catalysis fails to take place.